Hello, how nice to see you again. I hope you enjoyed today's practice. I'm not going to be using my teleprompter today, so please bear with me because I'm going to be working with a written text. Nevertheless, I will be stringing the terms that we reviewed 2 videos ago and giving them a context so that you can practice them while you are interpreting simultaneously. And for the purposes of today's drill, I'm going to start now. Ladies and gentlemen, I am delighted to be here with you today. Yesterday we talked about prokaryotes, and today we're going to focus on the eukaryotic cell. Eukaryotic cells are cells that contain a membrane-bound nucleus and organelles. This means they have a clearly defined nucleus and numerous membrane-bound organelles, including the endoplasmic reticulum, centrioles, mitotic spindles, Golgi apparatus, chloroplasts, and mitochondria. These specialized organelles occupy about half of the total cell volume, which means that they are much larger than prokaryotic cells. Another feature of these cells is that they divide by mitosis. What this means is that when the cell divides, it produces 2 new cells, 2 daughters or 2 daughter cells that are genetically identical to the parent cell. The genetic material of eukaryotic cells is organized in chromosomes, and they are capable of endocytosis, that is, of transporting large molecules or parts of cells or even whole cells into another cell. And this happens when patches of the plasma membrane pinch off to form membrane-bound vesicles that deliver nutrients from the external environment to compartments deep within the cell. Plants, animals, protists, and fungi all belong to this group. Now let's talk a little bit about the structure and function of these cells. The whole cell is surrounded by the plasma membrane that creates an interface between the cytosol and the environment. Their structure is complex and, as a result, comprises membranous and non-membranous organelles. Membranous organelles are enclosed within a phospholipid bilayer. And today we will be talking about the plasma membrane, the nucleus, mitochondria, rough granular endoplasmic reticulum, smooth granular endoplasmic reticulum, Golgi apparatus, lysosomes, and peroxisomes. The plasma membrane is a selectively permeable layer surrounding the cell cytoplasm. The nucleus is in the center of the cell, and it sequesters and replicates DNA, transcribes and splices RNA, as well as allows for selective exchange of molecules such as RNA and cytoplasm. Mitochondria are sac-like membranous organelles which perform aerobic respiration, are self-replicating and have their own DNA, and they are the storehouse for ATP, or adenosine triphosphate. And please note that the singular form for this word is mitochondrion. The rough granular endoplasmic reticulum is a network of membranous sacs called cisternae. Its rough appearance is due to the ribosomes that are attached, and by the way, ribosomes make polypeptides. The rough granular endoplasmic reticulum has the following functions: synthesizing proteins in the cell, making changes on proteins produced by ribosomes, creating new membranes in the cell. The smooth granular endoplasmic reticulum is a labyrinth of cisternae. Its functions are synthesizing lipids in the cell, transforming glucose into glycogen, modifying the nature of some toxic chemicals to reduce their toxicity, The Golgi apparatus is a system of flattened sacs that receive, sort and pack macromolecules for secretion and delivery to other organelles. Lysosomes are the primary components of intracellular digestion. They are small, rounded, membranous vesicles formed by Golgi bodies and containing a group of digestive enzymes. Finally, peroxisomes are vesicular bodies that are smaller than lysosomes and contain specific enzymes that oxidize certain molecules normally found in the cell, like fatty acids and amino acids. Non-membranous organelles. The cytoskeleton is the cell's internal scaffolding system, and it helps to maintain and rearrange the cell structure. It has different types of components, which are microtubules, actin filaments, and intermediate filaments. They can all be cross-linked by other proteins into networks or specialized organelles, among which we can find centrioles, which are small sets of microtubules arranged in a specific way and are involved in the formation of the spindle apparatus, cilia, which are short, hair-like structures that are used to move entire cells or substances around the outer surface of the cell, flagella, which are very long cilia, that is to say, long, hair-like structures that extend from the plasma membrane and are used to move for propulsion of spermatozoa. Microvilli are non-motile spine-like protrusions of plasma membrane supported by actin, and they increase the surface area of the cell. Ladies and gentlemen, I hope you have found my speech interesting. And as today's drill was very long, tomorrow we are going to take a break and we will talk very briefly about cell diversity. I hope you have found this video useful. See you tomorrow.